Municipal Code 57.60 still forbids smoking in this auditorium.
I dissent.
Tonight we will hear first from Dr. Edward T. Tyler on prospects for control of gestation. Gestation refers to the carrying of the embryo through pregnancy to delivery, the carrying of the pregnancy in the uterus. The new controls are of a number of different varieties, which Dr. Tyler will describe for us. They have to do with prevention of conception, the induction of conception, the changing of the locale of the gestation, and the influencing of the embryo during gestation. There are a number of implications to modifications in this field. When the fundamental processes of reproduction are interfered with, deep emotional conflicts are generated because these processes involve the essential sexual ties that assure species survival and around which all human communities have been organized. The unprecedented legal, moral, sociological dilemmas which these new techniques pose for us will be of concern to us in tonight's discussion and for many years to come. We are fortunate to have, as our discussant of the biological aspects of this topic, a member of the UCLA faculty in medicine and obstetrics and gynecology. Dr. Edward T. Tyler took his undergraduate work at Columbia, received his MD from New York State University, and has been associated with UCLA for many years now. He has a vast background in the fields of fertility, sterility, and related topics in obstetrics and gynecology. He is the president of the American Association of Planned Parenthood Physicians. He's the director of the Tyler Clinic, of which many of you know. We are also fortunate to have with us this evening Dr. Albert Tyler in the audience, a brother of our first speaker, who is known at Caltech and throughout the scientific world as an authority on the basic science aspects of the matters which Dr. Tyler will tell us about from a more clinical point of view. It gives me pleasure to introduce to you now a fellow adventurer through the pages of the Human Agenda, Dr. Tyler.
Thank you very much, Dr. Gurney. I'm afraid with that very nice introduction, what I have to say tonight may be rather prosaic, and I notice that a lot of you are missing the Academy Awards, but it's on television at the back of the room if you feel more inclined. At any rate, as Dr. Gurney has already indicated, this matter of control of gestation involves, in all its aspects, the control of human reproduction. And when we talk about human reproduction, as far as I'm concerned, we're talking about the most powerful force there is in nature, barring nothing, even atomic energy. And when we're talking about ways of controlling this force, we're really getting ourselves in quite a bit over our heads. And I think before I should start to talk about how we're going to control the forces of reproduction, we should probably enlighten at least a few of you on what we already know about reproduction. And it's really amazing how little many people do know about reproduction per se. We just are inclined to think intercourse happens, a sperm unites with an egg, and we have a baby, and that's all there is to it. But when you stop and think about all the developments that have taken place in the past, let's say, 50 years, you'll see that a tremendous amount of knowledge has really been accumulated. As a matter of fact, if you liken this whole situation to the situation that occurred with atomic energy, you will realize that perhaps 30 or 40 years ago, Many people knew that atomic energy was going to be used for specific purposes, but very few people gave much thought to it, particularly as to what would happen. Now, I think the same thing is occurring today. Many of us who are working in the field know what we can do ultimately with reproductive processes, but we really haven't thought too much about what the consequences can be. And I think this is the basic purpose of our discussion. It seems to me it's the basic purpose of the series, and I hope that a lot of thought-provoking ideas will come out of all of this. Now, as a lecturer, I think I'm going to use the device of putting most of this on the screen so that we can keep together on what I'm discussing. And if I may have the first slide, since we're going to talk about basic reproduction, I think the first slide will illustrate some of the things I plan to discuss. Can we start? Thank you. We started with some kind of a bang. Anyway, I think I'm afraid that this, to those of you who are versed in this field, will seem extremely elementary. But I do think that for many of you who are not inclined to go into this at all, some of the basic precepts might be of some interest. Now, first of all, I think it's extremely important that we all recognize that the process of reproduction has its initiation in the same general area in both the male and the female. And this particular area, as I've indicated in this diagram, which I'm sure you can't read in detail, is the pituitary gland, which is at the center of the picture on top. That's supposed to be a schematic representation of the pituitary gland. Now, this gland secretes a tremendous number of hormones. And again, for those of you who are not familiar with what hormones are, these are rather complex chemicals that the glands secrete directly into the bloodstream. They're vastly different from any other glandular secretions in the body because since they get immediately into the bloodstream, they have tremendously widespread ramifications. And the pituitary gland of all the glands is probably the master gland in the body because of the variety of hormones that it produces. Now, right now, we're talking primarily about the anterior portion of the pituitary gland. And this secretes hormones that apparently are identical in both the female and the male. And if you start on the left side, you'll notice the arrows that I drew are going to ultimately go to the testes in the male, and on the right side to the ovaries in the female, indicating that the hormones from the anterior pituitary, known as gonadotrophic hormones, are hormones that are stimulating each of these organs. And to carry this step a little further, I'll go into exactly what happens in the male and then in the female. But I've mentioned a few points here in the chart that we'll have to talk about a little later. First is this matter here of the basal temperature. I think I'll have to walk over it. This graph here. Most of you know about the basal temperature chart, and I think you're aware of the fact that if a woman will take her temperature every morning at the resting stage and do it conscientiously, during the first half of her menstrual cycle, she will have a relatively low temperature. When ovulation occurs and the egg is released, the temperature becomes elevated and remains that way unless she has a period. If she has a period, the temperature still will remain elevated. And actually, one of the most readily diagnosable signs of pregnancy is this persistent elevation in the temperature chart. We can say almost with complete assurance that if a woman has a temperature chart where the elevation has persisted for more than 14 days, she's undoubtedly pregnant. And I would say that's probably the earliest pregnancy test there is. Now, I've also noted here that this shift in the temperature occurs very near the time of ovulation, but most doctors as yet are not convinced as to whether it's just before or just after. An interesting thing, and when you stop to think about all the interesting things in the process of reproduction, it's really amazing. One of the very interesting things is that when sperm are deposited in the vagina, they meet a medium in the vagina that's quite acid. And ordinarily, the sperm cannot survive well in an acid environment. On the other hand, just above the vagina is the opening to the womb, the cervix. The secretion of the cervix is definitely alkaline under normal conditions. Now, apparently, nature has devised things in such a way that the acidity in the vagina serves as a protection for the vagina and keeps down infection and enables the normal bacterial flora in the vagina to exist. But at the same time, the change between the acidity of the vagina and the alkalinity of the cervix is undoubtedly one of the mechanisms whereby sperm will head toward the cervix and then up into the uterus. In other words, it's another assurance for keeping the process of reproduction going. We have sperm in an acid, unfavorable medium, desiring to get into the more favorable medium so that they don't stay where they would do no good. So this, of course, is an extremely important consideration when we're dealing with things like contraception or improving fertility. Now, another thing that I've mentioned here, and I think we may touch on a little later, is the matter of concentration of sperm. I'm sure that a number of you here know about the vast numbers of sperm that are present in each ejaculate, but if you don't, the average male ejaculate contains roughly about 500 million sperm. Now, when you stop to think that about 500 million sperm are ejaculated just to have one egg be fertilized by one sperm, this is really a phenomenal situation, but it's what actually occurs. And we know in sterility practice, for example, that if the numbers of sperm per ejaculate are much below 5 or 10 million, the individual is virtually sterile, even though it only takes one. So these are very, very important considerations when we're dealing with this entire phenomenon of reproduction. I've mentioned here in one corner the matter of immunological considerations, because this will lead to our discussion briefly of the sperm antibody reaction, the question of whether ultimately individuals can be vaccinated against sperm. Now, another very striking thing, again, I would simply like to state, because it shows how all the reproductive processes are so keenly correlated, is the fact that in the cervix, which is, of course, the base of the uterus, the secretions vary during the menstrual cycle. And it is only at the time of ovulation, when the egg is released and the woman is fertile, that the secretions in the cervix are favorable to the sperm and will help the sperm move through them. At other times of the month, if you look at the mucus, the secretion of the cervix, you will find that they will obstruct sperm. Now, whether or not this was intended to avoid having sperm have access to the egg after fertilization, I don't know. But at any rate, it is a very startling, as far as I'm concerned, phenomenon that this is the one time in the month when the egg can penetrate properly. Well, so much for this slide, and I think in the next one we go on a little bit further. The next slide, please. Drawing the lines over to the male, as you'll notice, I indicated the arrows stimulating the testes, which is in the upper portion of the screen. We have two different hormones from the pituitary gland which affect the testes, one of which is affecting the hormone-producing cells, and the other is affecting the sperm-producing cells. Now, this is an extremely important distinction, because there are these two bodies of cells in the testes. One is primarily responsible for the production of the male characteristics by producing these chemicals, and the other is responsible for prolonging the species and for spermatogenesis itself. The interesting thing is that you can have a disturbance in one part and the other part can be completely normal. As a matter of fact, in passing, I might mention that most sterile men have no difficulty at all with their male hormone production. The entire difficulty is with the sperm production. And these men are perfectly normal in every way except for the matter of producing sperm. And I think, unfortunately, in our society, there's a great deal of misinformation about this, and men who are not able to have children sometimes have stigmata because it's felt that they're not completely virile. Yet certainly the most virile men alive can have maximum virility and yet no sperm. And I think we have to keep that in mind. It may be a consideration when we're talking about some of the procedures we can use in correcting fertility. The next slide, please. We don't have time to go into all of this. This is a rough diagram of what we know about the normal menstrual cycle. We have these three hormones from the anterior pituitary gland, which we call follicle-stimulating hormone, luteinizing hormone, and luteotrophic hormone. And during the first part of the month, if this is the ovary and this is the lining of the uterus, we have the pituitary gland stimulating the ovary in such a way that the egg, which is implanted in a follicle or a sac, develops up to a point where it has reached maximum size and reached the surface. At the point where it has reached a maximum size, the second hormone, the luteinizing hormone, is released, and that triggers ovulation, so that the egg comes off from the ovary, it floats around temporarily in the pelvis of the female, and then is very shortly either trapped or finds its way somehow into the ends of the fallopian tubes, and then down from there to the uterus after it's fertilized. But again, here's another very astonishing thing that we've learned. At the place where the egg ruptures out of the ovary, there is bleeding. And this bleeding, of course, forms into a clot. Now, anywhere else in the body, if you have a clot, you're going to have some scar tissue. But in this particular situation, and I think it's the only situation in the body that I know of, instead of having a clot form, which is ordinary scar tissue, the place where the clot has formed organizes into a new type of tissue, which we call the corpus luteum. And this in itself is a gland. It has the same function as the ovary itself or any other gland in the body of this endocrine type. And for the rest of the menstrual cycle, this corpus luteum functions, as I say, as any other gland. If pregnancy occurs, the gland is maintained. If it doesn't occur, it disintegrates that cycle and a new one is formed the next cycle. The secretion of the gland primarily affects the lining of the uterus. It has other effects, but from the reproductive standpoint, that would be the thing that we'd consider. Now again, trying to bring to you this real miracle that's going on, while the pituitary gland is stimulating the ovary and producing this egg, we have it simultaneously causing the sac or follicle to produce the female sex hormone, which we call estrogen. Estrogen acts on the lining of the uterus, which has been pretty well shed during the previous menstrual cycle, and builds it up. It builds it to its maximum thickness, but it builds it to a point where it really would not be a successful endometrium for implanting an egg. What is required is that when it has reached maximum thickness, the secretions of the corpus luteum have to affect it and have to change it into a structure that will nourish and implant an egg. And the first part of the month development will not do this. It's only when it's had the addition of the progesterone from the corpus luteum that you get this. So here you have this very intricate sequence of events that has to take place in order to develop an endometrium that will receive an egg and maintain it after it has been implanted. Now, in the next slide, I think I show a little bit more about this sequence of events. Again, I told you this was going to be rather prosaic. We're not getting into anything sensational as far as I'm concerned. But at any rate, let's think of what happens with the ovum after it's fertilized. The ovary is represented up in the top of the picture. And again, we have the stimulation from the pituitary. We know that when the egg is released, as I mentioned, it floats around for a short while and then gets into the fallopian tube. When it gets to a point about a third of the way down the tube toward the uterus, it waits there, we believe, or at least it may not be waiting, but there may be sperm waiting for it. But there are sperm present at about that juncture. And the sperm will surround the egg. And astonishingly enough, since the egg ordinarily has a coating of cells around it, the sperm apparently carry chemicals in their cells themselves that will help dissolve the coating around the egg. The egg would not be penetrable by the sperm unless this coating of cells, which is rather thick, is denuded. So that perhaps we need a substantial number of cells to surround the egg to enable at least one sperm to fertilize. Now, the interesting thing here again is that once one sperm fertilizes the egg, a chemical change immediately takes place. My brother may check me on this, but this is my information. A chemical change takes place, and the wall of the egg is no longer penetrable by any other sperm. And obviously, this is a protective mechanism on the part of nature, because if we had more than one sperm fertilizing the egg, we'd have an odd number of chromosomes, and there'd be all sorts of problems. So the egg protects itself once the sperm is entered by preventing any other sperm from going in. Now, again, once the egg has been fertilized, it travels down the tube, and this process of tubal migration probably takes about 3 or 4 days, and then it finally winds up in the uterus. Now, here again, there's an astonishing phenomenon. As the egg is developing and traveling down the tube, it has to go into some kind of activity where it produces a chemical that will enable it to burrow its way into the lining of the uterus. The egg just doesn't get to the womb and sit down and live there. Something has to take place where it gets into the lining. The lining, after all, is a pretty solid structure, and we have to have something in the nature of an erosion type activity for the egg to get there. Now, according to normal circumstances, it takes about 3 or 4 days for these chemicals to be secreted from the wall of the fertilized egg. And so that usually, when the egg gets down into the womb, the chemical is being manufactured, and it therefore burrows its way into the lining, which has been prepared by the other chemicals that I mentioned. Now, if for any reason the chemicals are manufactured earlier, it would be very possible that the tubes would be the site of implantation. And as you know, in some circumstances, eggs do implant in the tubes, and we have ectopic pregnancies. We also have other types of pregnancies too, abdominal, that are on other bases probably. Now, the reason that some of these ectopic pregnancies occur, I'm sure, is because the tube has been damaged to some extent, and the egg isn't traveling down it at the rate that it should. And the egg itself has probably released these enzymes at the right time, but it just hasn't reached the uterus as yet. Now, again, another astonishing thing, and as I said, everything here is astonishing. At the site where the egg implants itself, a new structure forms. At the beginning, we call it a chorion. It's the place where the egg is uniting with the lining of the uterus. But this also becomes an endocrine or glandular structure. And the chorion, which is the combination of the wall of the egg and the wall of the lining of the uterus, starts to manufacture a hormone which is very closely related to the pituitary hormone. This is the hormone that doctors test for when they want to find out if women are pregnant, because it can be detected in the urine by a variety of means, including immunological tests and by rat and rabbit tests, and in very short order one can tell that pregnancy has occurred. At the same time, this particular hormone is responsible for stimulating the corpus luteum that I talked about and keeping it going throughout the pregnancy. The chorion ultimately becomes the placenta, which is an extremely important source of hormones throughout the pregnancy. And if it weren't for the placenta, obviously, in most cases, pregnancy couldn't continue. I say in most cases because there have been instances in animals where they can be sustained without a placenta. I think I neglected to mention that we believe that while the sperm may be capable of living for many days, we have a feeling that the egg can only live for perhaps 24 to 48 hours and can't be fertilized for much beyond 24 hours after ovulation. The next slide, please. I talked about the pituitary. The only reason I put this slide here is that for years we felt that it was a direct relationship between the pituitary and the ovary or testes, but in the past 5 or 10 years, we've all been convinced that most of the governing of pituitary action is from the hypothalamus, which is a group of cells at the base of the brain. And of course, if we recognize that the brain itself, rather than the gland in the brain, is the key to the situation, it explains such things as delay in menses when people have shock, or many types of pseudo-pregnancy, for example. These can also be explained basically on the fact that we have this hypothalamic center influencing reproduction. So I think, taking more time than I had hoped to, we've sort of summarized rather sketchily some of the things we know about reproduction, and it might serve as a framework to illustrate some of the things that we might do with this. Now again, I must emphasize that this is a very elementary type of review. Actually, when you get into each of these things, you really find yourself very deep into the matter of chemical changes of DNA, RNA, and all the associated factors that you've read about. So that, again, using this as a premise, I'd like to discuss very briefly some of the things that we might consider in controlling gestation, either in the very foreseeable future or in the distant future. The next slide, please. Now, here are some of the topics that Dr. Gorney said I had to discuss, so I've put them on the board. We have to consider in the future simpler methods of conception control that will be much more widely useful than even those that we have today. We must consider, as another type of gestation control, the achievement of multiple births, more than one infant at a time, on a voluntary basis. Then, I don't know, some people want twins. Then we have the fact that is already established, that of artificial insemination. And here we have two aspects: the solution of fertility problems in infertile couples, or the use of insemination for genetic or other purposes. Then we have a very interesting type of genetic or gestation control to think about, and that's the recovery of eggs, fertilizing these eggs in a test tube, which we would term in vitro, and then reimplanting the fertilized egg into the woman's uterus. Now, this could be an extremely important technique for those women who have tubes that are obstructed, where the egg cannot reach, or vice versa, either the sperm can't reach the egg or the egg can't reach the sperm. But there are a number of cases of infertility that are on this basis of tubal obstruction. And if they can't be corrected, as is very often the case, obviously if we could get the egg, fertilize it, and then implant it in the uterus, this could be a very important advance for those particular women. Another important gestation control would be the early detection and correction of embryonic abnormalities. For example, if we were further along in our knowledge at the time the thalidomide problem occurred, I'm sure we wouldn't have had the focomelia to the extent that it happened in Germany and elsewhere. So that early detection of abnormalities could be extremely important. Then we have the consideration of transplantation of ova. There could possibly be instances where women would want to have ova transplanted so that when they had relations with their husband, they could get pregnant if they weren't manufacturing ova on their own. The next slide, please. Now remember, I'm not advocating these things, I'm just telling you what can happen. Then we have a relatively related type of situation, and that's the recovery of eggs from one female. I think we get a little more obscure as we go along. But this one is the recovery of eggs from one female. I don't think he wants me to read it. Having it fertilized, and then subsequently having it developed in the uterus of another woman. In other words, the original woman is having the fertilized egg, but for some reason she can't carry it, and she's letting another woman carry it. I don't know. The other woman's womb is better than hers. I think we would call this a sort of womb service. Well, if you hadn't laughed, I'd have blamed it on our chairman.
Law professor's jokes.
Now, obviously, another important place to think about gestation control is in the latter stages of pregnancy, if there are requisites for surgery on the infant that seems to be developing improperly. Another point that I'll talk about briefly is the development of human embryos in artificial wombs. Then we have this very sensational type thing, the matter of parthenogenesis. And that's the development of an egg without the use of a sperm. In other words, the egg can be made to multiply and develop into a full adult animal by a variety of artificial means. And the male is not needed at all. Is this an advance? Now, I've indicated on the bottom here that all of these controls are theoretically possible. And in animals, some of them have gone quite a long way. But the important point is that if any of them are adapted to humans on a large scale, we're going to have to change a lot of our legal, sociological, and medical thinking, and moral thinking as well. Next slide, please. Now, as far as I'm concerned, I would like to talk mostly about the matters that will concern more people in general than those that will simply concern few people. And obviously the population problem is one that concerns all of us, and a very critical problem. You've been besieged, I'm sure, with information about the urgency of the population problem, so we won't take time on that. But I will say that at the present time, it seems to me that the recent methods of significance are the oral contraceptives, the intrauterine devices, and sterilization. Now, I include sterilization in that not because it's so popular in this country, but in certain parts of the world, for example, in India, this is accepted a lot more freely than other methods. As a matter of fact, there's been a lot more sterilization in India than there has been the use of oral contraception. Oral contraception in India is just on a very low scale of use at the present time. So these are the common present ones. Then we have prospects that might be available in the near future. I would suspect that before too long we'll have reversible sterilization. A male can get himself sterilized with the anticipation that if he ever changes his mind, as is not too infrequently the case, he can have this reversed. I'm sure that before too long we'll have injections that are available once a month, and they will work very much like the pills are working. As a matter of fact, right now, in our family planning clinic in Venice, we have several hundred women who are using this once-a-month injection successfully. The once every 3-month injection is also being used very successfully at our Planned Parenthood Center on 8th Street. So these, I would say, are going to be available probably fairly soon. We also have a real good prospect that there might be pills available that will prevent implantation fairly soon. Now, anti-implantation pills really present a problem, because if you remember what I discussed in the way of physiology, when you have a fertilized egg, for 3 or 4 days, it is not implanted anywhere. It's just moving through the pelvis. Now, if you devised a pill, let's say we had a pill that could be taken immediately after intercourse, and it would prevent implantation, even though fertilization may have occurred, you would obviously have another method of birth control. But you would also raise an extremely important question, morally and legally. Is the fertilized egg a life at the time the egg has been fertilized, or is it not a living being until it's implanted in the uterus? If it isn't a being until it's implanted, then this would simply be a contraceptive method. If it were decided that it is a new life as soon as fertilization occurs, then it would be an abortifacient, and it would be subjected to a lot of other considerations. So we have this particular thing that may not be in the too far distant future, because in Israel right now, Shelesniak is doing a lot of work with this particular method, and he seems to find that in animals at least it's extremely effective, and they're trying it in humans. We have a further point, which is early embryonic destruction, and that obviously is abortion because the embryo has been implanted, but there can be chemicals available that will destroy this embryo as soon as it has been implanted. This isn't the same type of thing as a late abortifacient, which may be used perhaps after the embryo has gone to 2 or 3 months, but it's one that starts out very early and would work only on embryos that haven't gone very far along in their cell division. This also is being tested quite extensively in Japan right now, because abortion in Japan is no problem, so that they can do these tests. But of course that raises an important question. Supposing you do have a chemical that can destroy the embryo, but let's say that in some cases the embryo is not completely destroyed but just injured. What happens if this fetus goes to term and is delivered and turns out to be terribly abnormal? Where are the responsibilities? Do you sue the drug company? Do you sue the doctor? Or do you sue the country for allowing the law to permit this? But it is a consideration that I'm sure Dr. Jones will discuss with us later. Now, of vital importance in our present culture is some method that will improve the use of rhythm, because whether we agree with it or not, there are vast segments of our population who will consider nothing for the control of conception other than the rhythm method. And it is very likely that with the advent of drugs that can possibly produce ovulation on schedule, we may be able to make rhythm a much more workable technique instead of the so-called Russian roulette as people describe it now. The next slide, please. Now then, we have prospects that are further along. First, the availability of a male oral contraceptive. For some reason, the husbands seem to get away from all of this. They leave everything to the wives. They have all the dirty work. The husband get all the pleasure. But at any rate, there is a very good likelihood that sometime in the future we will have a male oral contraceptive. Those that you may have read about so far in the papers are worthless at the present time. There is nothing that I know of on the immediate horizon that would work as a male contraceptive. On the other hand, I think you can realize that from what I described in the physiology, that we can have a male contraceptive that will simply work on the sperm-producing cells and will not work at all on the hormone-producing cells. And that, of course, is extremely important, because unless we can get one that's specific, only working against the sperm, we would run into the problem of virility, the ability of the male to have intercourse, and related problems. So we have to have a unilateral action of this contraceptive only on sperm. Now, something again that I'm sure will come before too long is the vaccination of males or females. In this, we would try to produce the same situation as occurs when you get vaccinated for smallpox. You have antibodies forming in the bloodstream which are hostile to the particular organism. In this case, the organism would be the sperm. And as a matter of fact, in some animals at the present time, they have been able to produce this immunity and avoid pregnancy. So that since we know it can be done in animals, we have to foresee that at some time in the future, it will be done in humans. Of course, I don't know about this idea of vaccination for preventing conception. It's bad enough now when you go through customs and they ask you for the smallpox vaccination. Suppose they start asking for your anti-pregnancy vaccination. But at any rate, it's one of the things that could happen. Now, again, this last item is an extremely controversial type of thing, and one that we have to anticipate as a possibility. We can have anti-fertility chemicals, I'm sure, that can be added on a mass scale to foodstuffs, to water, to anything that people will have common contact with, and these can prevent conception. At the present time, of course, we have nothing of this type, but the scientists who are doing work in the field are quite confident that this will come. But if it does, and when it does come, we have to consider the overall problem of whether or not government or whoever will be managing this won't be stepping in too far in individual freedom. I don't know whether in democratic societies people would stand for anything where their fertility is controlled on the basis of either this or anything that is not voluntary. And this, I would imagine, would certainly have to be an involuntary type of thing, because there'll be a lot simpler voluntary methods. Next slide, please. Now, again, a gestation control that's important to many people is the use of artificial insemination. At the present time, this procedure is used mostly in cases where the husband has either no sperm or is markedly deficient in sperm. And the simple procedure is to use a donor who's either a medical student or an intern or a PhD in one of the sciences who supplies the sperm, and these are simply... You mean we didn't make our selection so good, did we? At any rate, these are the donors generally, and it's simply a matter of applying the sperm to the cervix at the base of the uterus and having conception occur. A much more advanced form of insemination involves the use of sperm banks. And actually, while I know Time had an article very recently on the use of frozen sperm, this at the present time serves no particular purpose except for the convenience of the doctor. If he has a woman with certain characteristics and a husband with certain characteristics, and he wants to make sure that there are sperm available at all times, he can keep stored at minus 196 degrees centigrade samples of sperm that can simply be used at will when necessary. It obviates the necessity for a student or an intern or anyone else to come to the office at a given time when the woman is fertile and leave a specimen. And it's mainly from practical considerations that the use of sperm banks are important at the present time, but not for any necessary reason. And incidentally, I would say that at the present time, probably in the California area, more cases of pregnancies with frozen sperm have occurred than anywhere else in the world. I think this particular technique is more advanced on the West Coast for some reason than anywhere else. And as I mentioned, it's largely for convenience. However, if we recognize that you can preserve sperm, you also have to recognize that it has very far-reaching ramifications. For example, we could take sperm now and store these sperm for up to, well, at the present time, I'll have to say up to 2.5 years, because that's the maximum that we've done it with human sperm. They've gone a lot longer with animals. But with humans, we know that they can be saved for 2.5 years. Now, this, of course, will be increased later on, and I would be surprised if in the next 30 or 40 years, or even less, we're not at a point where sperm can be preserved almost indefinitely. And it has genetic possibilities. It's also extremely important when you consider that these can be stored in lead containers or other containers that would be foolproof against radiation. And we would always have a source of normal sperm for subsequent fertilization. But to make it complete, we'd also have to have eggs stored, and this can also take place. Because even though the eggs, as I mentioned here, are not as readily amenable to this type of treatment, I'm sure before too many years go by, they will be. So that the use of frozen gametes, as they call the sperm and the egg, we can anticipate that conception can take place long after the individuals are dead and gone. Of course, practically speaking, you might have a case where, for example, a soldier goes abroad and leaves some sperm behind, and when his wife is ready to get pregnant, she can get pregnant. The only problem is, how is she going to explain this pregnancy to her friends if he's been gone for 3 years? The next slide, please. This simply illustrates the type of sperm bank that's used. It's a liquid nitrogen container, and as you'll notice, the temperature is down to minus 320 degrees Fahrenheit. They have to be kept at very cold temperatures. They sort of hibernate at that temperature. The next slide, please. Next slide. Now, another control that I touched on at the beginning is this matter of multiple ovulation and achieving multiple pregnancies, twins, triplets, and so on. You've all read in many places about the production of quintuplets, quadruplets, and so on with the new pituitary hormones that are available. Now, very simply referring to this, these hormones of the pituitary cause the release of more than one egg. Since the pituitary governs ovulation, if you give human pituitary preparations, you can cause the release either of one egg in women who are not ovulating, or perhaps even more eggs in women who are ovulating but need extra stimulation. The only problem at the present time, as far as producing multiple births at will, is the fact that at our present stage, we have no real way of controlling dosage or knowing how many eggs are going to be released. But since we have the products and we're working with them, I would be quite convinced that before too long we will have voluntary production of twins, triplets, and even quadruplets if people are silly enough to want them. I say silly enough because the incidence of carrying to term with triplets and beyond is very low. They present a real problem. The next slide, please. Now, something again that I think is of importance to a lot of people is the matter of deciding in advance the sex of the offspring. At the present time, there have been many experiments which have indicated that you can separate the sperm that are responsible for carrying the Y chromosome, which is responsible for the male. And these sperm, that is the male type sperm and the female type sperm, if we can use that expression, are identifiable microscopically. And I think Dr. Carlson mentioned last week that there's a pretty good chance they can be separated by electrophoresis and other methods. So at any rate, we do have to recognize that before too long we will be able to separate these male type sperm and female type sperm and use them for producing one sex or another. As a matter of fact, I think it's probably easier to detect male and female type sperm right now as compared to, with the present haircuts, male and female type people. So at any rate, we do have this definite potential. There is another theory on predetermination of sex which involves the timing of intercourse, so that in certain instances a male will be more likely produced and in others a female will. Now, I think this is a very controversial matter at the present time, and the investigators who are involved in it are not unanimous as to which time of the month each one would be more likely. So I feel, as I've indicated on the last line, that ultimately, if there is real desire for a male or a female, this will be on the basis of separation of sperm and the semen and insemination with that particular group of sperm. The next slide, please. I think I explained this enough initially. All I would add to this is that at the present time in Japan, a doctor by the name of Hayashi feels that he's gotten quite a bit along in this particular procedure. He's been able to fertilize human eggs in the test tube. He's extracted them from the ovaries in these cases of tubal obstruction, and he's reimplanted them, but unfortunately, with the present techniques, they haven't developed very far. But I think, again, it's conceivable, a term I shouldn't use, but it is conceivable that before too long, we will be able to do this particular technique properly and treat sterility in a number of women who otherwise would be barren. The next slide, please. This, I mentioned earlier, of course, very simply, is recovery of the egg from one woman, having it fertilized and then implanted into another uterus and carried to term by the other woman. Now, this isn't as ludicrous as it sounds, because there are many cases now of RH difficulties where the woman who's manufacturing the egg has been immunized, has antibodies which would affect the embryo and which will not necessarily be detected in time, so that you may have a damaged fetus, whereas if this fetus were carried in a woman who was not immunized, the chances are very good that she would go to term and deliver a normal baby. But again, this brings us into legal and moral and sociological problems that I'm going to stick Dr. Jones with. The next slide, please. Now, then we have this matter of fertilizing the egg and maintaining it in vitro. I mentioned already that this has only been done on a very limited scale. It's been done with rabbits, which of course, since rabbits are mammals, we have to assume that ultimately it will be done on a larger scale with humans. Once they're fertilized, they can then be introduced into artificial wombs so that the mother will not have to carry the child. And whether this is good or bad, I don't know. This, again, is something we'll leave to Dr. Jones. The next slide, please. I think I've illustrated. The next slide. Here, for example, you may recognize this. The best picture I could find on this type of thing was in Life. You know, that's the modern scientific journal. At any rate, this simply illustrates an early abortion where the placenta and the embryo were expelled intact, and they were then put in a pressure chamber in physiological salt solution, which had a high tension of oxygen. And the idea here was that the embryo would receive massive amounts of oxygen and survive by virtue of the pressure chamber and the normal solution in which it was immersed. And in this particular case, as I recall, the fetus only survived perhaps a few days, but it's not beyond the realm of belief that techniques can be available that will keep these embryos alive a lot longer. The only problem medically that I see in this is that a vast majority of early abortions are caused by abnormalities in the fetus. And you would then be confronted with a problem of whether or not you're saving a fetus that would ultimately be defective, and you'd also be confronted with a problem of trying to decide whether or not a given fetus should be saved. In other words, to use the expression that's been used many times, the doctors would have to be playing God to decide which embryo should be carried further in its gestation. But it is a very possible approach in the future if it is desirable. Since people are working on it, we have to assume that ultimately it will come to pass. The next slide, please.
Dr. Tyler, in view of the fact that we've come to the edge of the separation of church and state, I think we should get from the poetry of this presentation back to the prose of human values pretty quickly.
Well, I thought Dr. Jones would take the prose of human values.
I thought you gave me the poetry. Every time I've heard you say I'm going to talk, I thought it was poetry.
Okay. Well, shall we call it poetry or prose?
You take the prose, I'll take the prose.
Okay. As a matter of fact, with about 2 minutes left to go, I don't think I'll have time for either poetry or prose. Anyway, this is carrying it a little bit further using an artificial womb with a placenta. In the center of this, we have a placenta which is attached to a fetus, and on the bottom, as is in the next slide, may I have it? We have the plastic replica of blood vessels that go to the mother. And obviously, this use of an artificial placenta is something that could, in the long run, be very practical. I can conceive of patients with toxemia of pregnancy who should not continue their pregnancies and who would require something of this type that will carry the baby to term for the last 2 or 3 months. The next slide, please. Obviously, we all know that surgery on the fetus is currently being performed. We have advanced animal experimentation. We know that in certain RH problems, we're transfusing the fetus while it's still in the womb. And if we're doing that, we have to consider that ultimately we'll be able to get at the fetus itself and correct any congenital defects. And some of these obviously are much more readily correctable during pregnancy than after, while there's still development going on. I think the next slide illustrates how they've been able to do this. This is an illustration of a monkey fetus where it was removed from the womb for approximately an hour and was subjected to a variety of tests and given various chemicals, then reinserted and went to term, and the mother of an infant monkey and the monkey itself had no ill effects. Now, obviously, if we can get as far along as monkeys with something like this, we can assume that it won't be long before human fetuses can be withdrawn from the uterus, given medication or chemicals or anything that the fetus might need, reimplanted and then delivered later on. So this isn't too far-fetched, and there might be specific instances where this is required. The next slide, please. Then this is really a sort of an ethereal, this is a poetry type thing, the use of parthenogenesis. I would say immediately that as far as humans are concerned, I really don't see a place for parthenogenesis. But in any case, it is the development of an embryo with no fertilization, and the sperm has not had access to it. We know that it was done for many, many years in lower forms, and in fairly recent years in mammals, meaning rabbits. And predominantly the characteristics of this new being would be maternal, but not necessarily so. As a matter of fact, my brother here insists that even though this might be an egg, you could still have males resulting from it. Maybe if we give him a couple of minutes, he'll explain why we can get a male. I know I didn't understand it, maybe you will. But at any rate, it can be done. Then we have this matter of genetic consequences, which could be extremely serious because you don't have the intermingling of the chromosomes. You have just the chromosomes from the mother, even though you have characteristics from antecedents, but they're still unilateral. And obviously, the social consequences would be very serious. You know, we have a matriarchal society right now. Think about what happened if this took place. Sometimes I think science should just sit down and stop, and this is one of the places. The next slide, please. This is on the same subject, and it indicates that at least in some species of plants, and also we know now in animals, you can simply take a single cell of the mature individual and implant it in a nutrient medium, and finally transplanting it to other media where it will grow properly, you can get a full replica of the mature individual. So that this doesn't relate to the sperm or egg cells themselves. This relates to any cell in the body. And this would, I would say, be a very astonishing type of I don't know what I'd call it, but it would be astonishing. I just think this, again, is one of the really far-fetched types of approaches that science is looking into today. The next slide, please. I don't think we have to spend much time on this because it simply relates to organ transplants, but as I have to mention this because Dr. Gorney is really interested in this, you can have individuals, as he and I have both seen, where they've had hysterectomies. They still have their ovaries, but for some reason the uterus has had to be removed, and therefore they could not carry a pregnancy. If you can take a uterus from someone else who doesn't need it and implant it in the first individual, you know, a lot of women don't need their uterus. As a matter of fact, they'd prefer it without them. If you can do this, you can have a fertilized egg, have it in a uterus that's been transplanted, and it can go to term. Now, this is not at all extremely far-fetched because, as you know, the first usage of transplants to any large extent was taking the aorta from dead people and transplanting them into individuals who had aneurysms. And these were life-saving measures. And these transplants took. Right now, a lot of this is being done with plastic aortas, but at the same time, we know that these structures can be transplanted, and if they can be, then we can have this type of usefulness. It might be a little less essential to have transplants of ovaries, but this would also probably be done. And it would be done when the ovaries are defective. The big issue here is not the same as the one with transplanting the uterus, because here you're transplanting the entire hereditary characteristics of the individual. And you're not dealing with a woman. The first woman obviously would not have the characteristics of her heredity in the child. It would be the other woman's heredity that would carry over. But again, this can be done. The same thing, I'm sure, can take place with transplantation of the testes. Do I have any more slides? I hope not. I think that's the last one. Is there anything after that? No. Well, we ended with a bang. I think, going through all this, I think the thing I would like most of us to carry away is the fact that all of these things are going on, and as long as they are going on, it is not too early right now to think about what might be happening if they do come to pass. We made a mistake by not thinking enough about the A-bomb and the H-bomb when we knew it would ultimately happen, and let's not have the same thing happen with our reproductive processes. Thank you very much for your attention.